Friday. I really thought I was on the right link. And like, I guess I had like pulled it up, but like when I clicked start, it was like on a different part of the screen and it was starting like a new thing. And so I like recorded it on a computer. And I was like, kept being like, why is nobody joining? Why is nobody joining? And then I got off and I got all these emails saying people had joined. And I was like, oh, great. Like that's my B. So I don't know. I was on my iPad, so it was a little bit different from what I was used to, but everything's a learning curve this semester. It just seems like it's one thing after another lately. But anyways, we're gonna, like I said, pull up the Kahoot stuff. But as far as I'm bringing it back to stuff that's like in the now, as far as it goes with now of course I completely lost my train of thought. the top hat stuff, right? So we closed off top hat, like I said a million times, and and we switched our pre-labs and our in-labs, and the post-labs are already on Canvas, right? So now everything's on Canvas. There, if you go to that lab assignments, documents, and worksheets, I added assignments in there. That first link on there should take you to all of the assignments for lab, and it'll like be a page for each kind of assignment. So you can go and find, okay, if you need to, if it's not popping up or populating in your to-do list, your by date assignments, whatever, then you can find it there. I think, like I said, I think a lot of my online people don't understand that they don't have to sort it by date, their to-do list or like their assignment list. You can sort it by type, because those in-lab pictures that we take to upload onto Top Hat originally, now we're uploading it onto Canvas, which a lot of you have figured out. You can find those by sorting your assignments by type, because they're not going to be due till October 15th. So they're not going to pop up on your to-do every time we have lab. That's just because I didn't think there's any rush for uploading the pictures. You know, I didn't just get them on there at some point, please. So get them onto their respective thing when you get to it. But all right, now this is. But are you guys liking that better than Top Hat? Yeah. Thank you. Okay, that's what I was the most worried about. I was like, I worked my butt off to get it onto Canvas. Like when I had enough, I had you guys complaining about it, and I had my online people, and then my Tuesday morning people. And then I was like, you know what? I'm not doing this Top Hat thing. And I haven't had quite as many issues before with it in my past semesters, which happens. That just happens. I don't know why, but it does. Some semesters are just different than others. So I didn't want everybody, you know, to keep having to suffer with it. They didn't have to, so I put everything onto Canvas. But it's not as simple as just like putting it on there and, you know, happy times. I had to write all those pre-lab things. I literally wrote that Thursday in like one afternoon. So if you come across a question that you get wrong on those, because those do show you like your results on those. If you come across one that you got wrong, I've already found one and I corrected it, but let me know. And I will go back and look at it and see if it is in fact wrong. And I just didn't mark the right thing, because I was just didn't mark the right bubble when I'm selecting the answers. That's usually the issue. So, but yes, it should mostly be fine. I was trying to work really hard on those. I've got all of them up now for the entire semester, and all of that. So we should have most of our resources there available to us. Easy to just go through. Those will pop up automatically on your to-do lists, and so you can get those done now. And you don't have to go to Top Hat to do it. And your grades will populate automatically, and I don't have to type them in manually, so I'm happy with that also. Also, in the lab, we're going to get to lab. We'll talk about it when we get to lab. Never mind. Okay, so C5, really quick. Let's do it. 5. Okay, so we'll go over these always in the classic mode, where I will go through the questions with you guys, but the lab ones will be the games. Yeah, they're really cute. Yeah, I need to get some stuff with Pistol Pete on them as everything, like, or like OSU stuff. I have orange, but I don't really have like, no, the OSU stuff. Yeah, to be clear, like when I complain about how back in my day we did blah blah blah. I was at OSU Stillwater, so it's not like I was at some sort of crazy school where they did things different. No, it was OSU Stillwater. I took this class at OSU Stillwater, so 20 years ago though, not recently. A long time ago. So we'll get started. Remember, you can always join in if you come in late. I had a laptop, but it's not people brought them to class. They didn't really have good battery life back then. Which of the following is found in a eukaryote, but not in bacteria? It's the main feature associated with eukaryotes versus bacteria and archaea. Kind of in the picture too. It kind of says it. It's an old word for it, but. All right, so it's going to be the nucleus. Exactly. Remember that karyo. If you guys have ever heard of karyotyping, that's whenever they sort out the chromosomes, make sure you have the right number of each one. Well, that's associated with the nucleus. So that comes from. The genetics thing though. So this organelle turns chemical energy into the cell's energy. Yep, you guys got it. Mitochondria, that one's not too bad. It's one that I think a lot of us have heard of before. So. What is the function of the endoplasmic reticulum? Yep, so especially the rough ER, right? So the rough ER is the site of protein production. That's where we have the ribosomes studying the membrane of the rough ER. Faculty Senate. All right, what are the organelles that contain chlorophyll? That is chloroplast. I like that it's kind of in the name. I like that you guys are getting the right answer on this. True or false? Yeasts are fungi. So the fact that we have them here in the eukaryote chapter, we know it's a eukaryote, but is it a fungi? All right, so they are fungi. You guys got that right. They are a type of fungi. I know we always think of molds and stuff, but yeast are too. So true or false? Fungi have chloroplasts and they can perform photosynthesis. So this is a tricky one. We certainly didn't really talk about whether the fungi were going to perform photosynthesis that much. So good. I'm glad that you guys were in on that. So that they do not perform photosynthesis. They do not have chloroplasts. They are not plants. All right. Do algae help to provide oxygen to the atmosphere? All right. Good job, guys. So in the kingdom Protista, the subkingdom algae, these guys are the ones that provide most of our oxygen in our atmosphere, probably since the beginning of time. They're probably the ones that like changed our atmospheres. All right, these single-celled organisms have trophozoite and cyst life stages. All right, so that is in fact protozoa. I'll say real quick for the protozoa stuff, if we're talking about infections that are caused by protozoa, remember the cysts are going to be like the dormant hardy ones, right? They're going to be the resistant ones that are there to survive an environment. And the trophozoites, like I said, think of eating out of a trough. If you're eating, you are not dormant. So they'll be the active and reproducing ones. But when you get infected, typically, if you are a cyst forming protozoan, not all of them do this, okay? But if you do this, it's typically the cyst that's going to get into your body and then, initiate the infection. But the thing that's actually leading to causing the true infection itself and then giving you symptoms and everything, that has to be the trophozoite, because the cyst can't multiply. So the cyst gets in there, but the trophozoite is the multiplier. So if it's doing a thing, then it is the trophozoite. If it's doing a thing, then it is the vegetative cell for the bacteria, right? If it's just chilling, no. So what are helminths? I feel like the next one is a term. All right, yep, these are our parasitic worms. Good job, and they fall into kingdom Animalia. What are platyhelminths? Or platyhelminthes is the term for it, but we call them platyhelminths. All right, so these are the flatworms, and again, it just sounds like flat to me compared to any other thing. So that's why I remember it that way. All right, good job, guys. We'll get our little runners up. All right, and then let's get started. Get out of here, dude. I guess you should close this whole thing. Yes. All right, let's get into viruses. So full disclosure, when I was in graduate school, I studied mostly viruses. When we get into immunology, you'll hear my immunology story. It's not really a story, it's a statement, but yeah, I studied mostly viruses. So that was like my focus. Set it. Watch it make me sign in. So have me sign in probably, and then also do. Yes, we'll go over this because we got the test coming up. I usually like to go over a question that might be on the critical thinking end. So not all the questions are like this, right? So this is one of the critical thinking type of questions that we might have on the exam. There are definitely more than one, but it's not necessarily all of them. So I wanted to go through one of these with you guys to make sure that we can think through the answer to this one together, right? All right, 36-year-old man presents with diarrhea and abdominal pain. Just a few hours before, he reports having eaten at a new Chinese restaurant. The only thing he ordered was fried rice, and he stated that it tasted like it had been sitting under a heat lamp all day. Eventually, after a few more cases pop up, an epidemiologist is sent to the site to investigate. The epidemiologist discovers that the fried rice is contaminated with gram-positive endospore-forming rods. What is the most likely cause of the illness given this information and these choices? So our choices are Salmonella, Bacillus cereus, E. coli, and staph. All right, so let's see what we can remove. You might be looking at staph and be saying like that doesn't cause diarrhea, so we're out. But it's actually the #1 cause of food poisoning. If you go to like a family event and eat like they warn you about, don't eat the potato salad that's been sitting out for longer than an hour, and don't leave your food out for an hour, it's typically staph. Staph produces a toxin, and so it's true food poisoning. You're not getting sick from the bacteria, but from the toxin that it can produce. And so it's actually pretty quick effects. You can eat something like that and get sick within 30 minutes, 30 minutes to 6 hours. So wide window there, but yeah, so we couldn't necessarily knock out staph from this other than the fact that what feature in this tells us that it isn't Staphylococcus aureus. The rods, exactly. So we know that it's rods. So automatically, that's one you can rule out just from the name alone, because those aren't going to lie to you, those names, right? because coccus is round. All right. So we know it's a bacilli of some kind. As it turns out, and you guys wouldn't know this necessarily yet, but all three of these, A, B, and C, they're all rod-shaped. So, yeah. so I'll give you that one, okay? So now we're dealing with gram-positive endospore forming. So there's two things we have going on there. Don't forget the endospore forming. So if you ever get something like endospore forming or acid fast or lactocell wall, those guys are going to narrow it down significantly for you guys. So one of the things that we learned about and that we have mentioned, and it's still new to you guys, but I'll definitely be bringing it up, especially when we get back to the endospore stain. Endospore organisms are always going to be, maybe you guys remember me saying this, bacillus or Clostridium. We talked about Clostridium difficile, which is C. diff. We talked about Clostridium tetani, and there was also a Bacillus anthracis. That's an obvious one, you know, anthrax. It's only ever going to be Bacillus or Clostridium. So that actually right there, just that one word, endospore, the fact that it makes endospores, well, you're only going to be looking for Bacillus or Clostridium in your choices. And so it's B. So if you know that information, which is on your study sheet, and you guys are able to recall that information, or at least remind yourself, okay, I remember that endospore is only a few things. Would it be Salmonella or E. coli? I don't remember talking about that. And we know coccus isn't a rod. Maybe that would help you, you know, cut it down. A lot of people, whenever they read this, they stick on to that diarrhea and abdominal pain, and they just say it's Salmonella because it's the first choice that they read. And we all know Salmonella causes those things, but literally all of these do. And so many more do as well. So having diarrhea and abdominal pain doesn't help you diagnose it. You have to get, you know, other circumstances around it, of course. In this case, we're getting information like there's endospores. So it has to be Bacillus cereus, specifically because endospores can only be Bacillus or Clostridium. So you're only looking for those two as the first part, the genus, right? All right, does that make sense to you guys? So you see, if I break it down like this, it seems simple. And I love you, but when you get into the test, you guys are going to freak out when you read questions like these. Just remember, just to read it through, maybe even make notes of what sounds important and see if you can break it down from there. Don't let it overwhelm you because it's a lot of words, but most of it's just words, right? So don't let it overwhelm you. All right. And I kind of do that on purpose because, well, the NCLEX does it too, but NCLEX does that. If you guys don't know, there's a whole part of it for critical thinking. And that's where I pull these ideas from. I don't pull the exact words, but I base it on those questions. They're meant to give you a whole lot going on. And then basically like one little question built into it so that you can see through the muck of all the other crap that's in there. Because if you guys are going to be going into healthcare, whether it is for animals or people, it doesn't really matter. But there's going to be a lot going on with your patient, and you're going to need to see what's important in it. And that's what these are kind of putting it in a scenario, just like you would see it if you were dealing with it. So that's the idea with that. Okay. All right. Viruses can infect every type of cell. If you are a cell of any kind, then you can potentially be infected by a virus. So do we consider them organisms? Are they really alive? What role do they play in evolution? And a lot of this stuff will actually be quite shocking if we really broke it down. Their distinctive biological characteristics, how can they be so small and so deadly? And what is the connection between viruses and cancer? Well, we know there is a connection between viruses and cancer. If you ever heard of cervical cancer, and you probably know that, HPV, having HPV is a cause, a literal cause, not a correlation, but an actual cause for cervical cancer. So it's something to be very aware of. And there are probably others out there. And, you know, nowadays scientists are high believers in the fact there's probably a lot of our cancer or other disease that we may have may in fact be caused by viruses. We just didn't know. We haven't looked for them. We haven't found them. How many times do you think you may have gotten sick in your lifetime from some cold virus that nobody's ever discovered before? It's probably happened. And that's crazy to think about. There's so many viruses out there that nobody even cares enough to study. So we haven't really discovered them. So, interesting to think about. Also, a lot of viruses introduce their DNA into your DNA. They literally insert their DNA into your DNA. And a very large, I think 30% of our genome, it might be more than that, but no, it's at least 30, it consists of old virus. Like they're not active virus, like, you know, HIV or something, but from ancient back in the day, and it's correlated with viral genomes and not with our own genome. They may not code for something, or they may. Just depends. There's a lot of trash in our genomes, but yeah, that's kind of terrifying to think about, and what roles that may have played in evolution, what genes it may have allowed us to develop that gave us ability to survive better. All right, so we typically will call viruses active or inactive, right? So we don't say that they were alive, and we don't say they were killed. Although if you go buy a bottle of Lysol, it'll say kills 99.9% of flu virus or whatever, or COVID virus or whatever. But yeah, we typically want to lean more towards active or inactive because of the components of viruses, which we're going to get into. Oh, and they are considered obligate. This is probably the most important feature if you're going to describe them as something. Obligate intracellular parasites. Sounds fancy. Obligate literally means obligated. If you are obligated to do something, you have to do it, right? So this guy has to be an intracellular, so inside of a cell, parasite. So viruses, all of them, cannot reproduce without being inside of a cell. That's what this is saying. Why are they parasitic? Because they harm us, they use our resources, they deplete our resources, that sort of stuff. So it's harmful. We don't get any benefit from them typically. Like we said, some of them have, there it goes, 40 to 80% of the human genome. Wow, that's even more than I even said it was. That's just nuts to think about. But yeah, they're part of our biome. There's probably bacteria that have their own viruses on your skin right now, all of that sort of stuff. So they're part of everywhere you look. We just don't talk about some of the ones that don't cause obvious illness a lot of the time. All right, so they are not cells. I mean, I guess basically we're going to go through a lot of these topics here in a moment, so I'm not going to go into it right now, but this is kind of a good little, I want to say table outline of features of a virus that you guys can come back to for studying. To put things into scale. This green thing is a yeast cell that is budding, right? It's a little bud coming off of it. Our cells, our skin cells and stuff are bigger even than that. So let's look at these. If we break this down, herpes, rabies, HIV, flu, adenovirus, this is a bacteriophage that infects bacteria, poliovirus, yellow fever virus. And then this little dot down here isn't a virus. This little dot down here is a hemoglobin molecule, which is just a protein, y'all. This is how tiny these guys are. So yeah, I guess yellow fever virus, you look at that, it's quite small. I always talk about polio. Now polio is one of the smallest ones, and parvo is as well. So those guys are just ridiculously small. We don't think about how small they are, but even compared to bacteria, All right, so these guys don't even resemble cells, really. So when we think about a cell, what do we think about? We think about a membrane-bound object that has its own genetic material, at the very least. Maybe its own ribosomes. It can do everything it needs to in order to make a copy of itself, right? It doesn't need to be stealing somebody else's things to do it. It can go from one cell to two cells all on its own. It's capable of reproducing. It's everything it needs. It's all membrane-bound. Viruses only need an external coating of some kind and its nucleic core. And what I mean by its external coating, we'll get into in just a second, but that's the basics. So it only really needs two things. There it is. So our outer coating that every single virus has to have is called a capsid. I'm going to leave these here. It's called a capsid. So the coating on the outside of a virus can be different from virus to virus. Sometimes they're just a capsid. Other times they're a capsid surrounded by lipid bilayer. How can they get lipid bilayer on them if they can't do their own things? We'll talk about that. But the absolute two things that you need are a capsid and your nucleic acid genome. Please be aware that I put nucleic acid here. I did not put DNA. Okay. All right, so the bare minimum is these. Some of them might have that envelope, which is different than the cell envelope. I'll always put a cell envelope when I'm talking about cells. These are not cells. Okay, these are viruses. So viral envelope, if you will. And an envelope is exactly what it sounds like. It's whatever envelops the thing. We're talking about a cell envelope, everything that envelops the thing. We're talking about a viral envelope, everything that envelops the thing. Okay, so it could be envelope. It's usually a modified host membrane. I'll just put membrane here. And then the other thing, spikes. So we always have spikes too in some form, but sometimes the spikes are just part of the capsid. Other times you have to have separate ones to go on the outside of the envelope. So let me show you what I mean by that. In order to get into a host cell, you need a key, basically. So viruses are carrying around a key in the form of these spikes, and they're glycoprotein spikes. So it's sugar protein, and they have a specific shape that fits into a specific block on the cells. So receptors or whatever that normally do other things on the cells. So these guys can come up to it, shove their spike key into it, and then get into the cell based on that. So that's why the spikes are important. And if you ever see viruses depicted and they always have those little spikes coming off, that is literally what we're talking about. That's why they're called spikes. So those are the proteins that will dock with the target cell and allow them to infect it.
But not all of them have spikes.
They all have. So this is the hard part, right? So let me real quick draw this. So if you are something that only has these two, okay, you only have capsid and nucleic genome, but that's the bare minimum that you need. All right, these guys are called naked viruses because they don't have the membrane, the envelope, right? So these guys can have crazy shapes and whatever. Let's just draw it like this. And then they'll have spikes coming off of them, but they're considered part of the capsid. They're not considered separate. Okay, so they still have the spike function. It's just the spikes are not separate from the capsid. For the enveloped viruses, now you're probably wondering, okay, well then how do these guys do it? They might have a similar capsid and nucleic acid. Like imagine the nucleic acid's in here. Okay, right. They might have this, but if they put spikes on the outside and then coat it in a membrane, how is that going to interact with the next cell, right? So they'll take their capsid, their own capsid, and their own nucleic acid. That will bud through and obtain a membrane on it that is already studded with spikes. Because we still also need to get back into the next cell. So these membranes that they have on here, they are your membranes. So if you have had COVID or the flu, then you've had this before. And they have membranes. So they'll steal your membranes from your cell because as they're building up, they just pop, okay, they get into your cell, they just pop their little nucleic acid into your cell, and your body just starts cranking out copies. It just sees it, it's ribosomes and whatever else. It's used to seeing mRNA and things like this. So it'll just start making out protein, reading it and making protein, making copies of it, because the proteins are then direct that you need to make copies of this. Also, don't just forget to, don't make copies, just protein, also make copies of the genome. Don't you remember a cell? This is what we're doing. It's a good idea. And that's kind of what the viruses will do. Hijack your machinery, hijack your cell's thinking to make their own copies. So we've got that cranking along. We've got all the proteins that make up the capsid. Remember, proteins are just going to come together and make up, they're like building blocks that we're going to build the capsid out of. The units that we build our capsid out of are called capsomeres, and they come together to make these weird shapes that we'll see. And they'll encase, the whole point of the capsid is to encase our nucleic acid genome. So we'll make our copy of our genome. We have our capsid proteins that are being made by the cell, and they'll come together just naturally. They're like naturally just want to be together, like we talked about with the hydrophobic and hydrophilic. That drives how these guys are going to come together, create these weird shapes. So we'll get this made here. This is called our nucleocapsid. That's our nucleic acid with our capsid, right? So our nucleocapsid. In this case, we have a membrane. One of the other proteins that we're making is spikes. It's included in the capsid for the naked viruses, but for our envelope viruses, they have to make them separately. So we're going to make our spikes and literally have the cell put them on the surface of the cell. That way, when this guy comes out that's inside of here, all it does is push through the membrane. Like imagine if you were like pushing through like a big old blanket. You're just surrounding yourself with a blanket, and then shoot, now you're surrounded with a blanket. That's what they're doing to your membranes. So we stud your membranes with that spike protein. and we push our way through the membrane, cover ourselves with that membrane, and therefore the membrane that's studded with the spikes. So they can go on and cause infection in another cell. Beautiful. Amazing concept when this is all they consist of. They don't have ribosomes, they don't have, mitochondria, they don't have anything. And they can cause massive damage to cells, right? Look at Ebola. Talk a little bit about how that happens in the future, but right. So here is a functional virion, much better than the pictures that I was drawing you guys. On the left is our naked virus. This could be something like polio that does not have an envelope, because not all of them have it, right? The bare minimum you need is your capsid, and that'll be around your nucleic acid. And here we're pointing out the spike, which again, it'll be part of your capsid, okay? But it's still a spike. It still has the same function. All right, whenever you have an enveloped virus, which is on the right, that guy will have our nucleocapsid, but instead of putting the spikes on it, we'll put it on our membrane and we'll just plug through the membrane and surround us with it. There's only two kinds of viruses here, naked or enveloped. And that's all you really need to understand for the structures of those guys, okay? Turns out though, there's different kinds of capsids that we can make. So the capsids themselves are just repeating units of capsomeres. They're just protein subunits that come together to make the capsid. And they will self-assemble. They just naturally will come together spontaneously. They don't need any help with it. So they'll self-build themselves. But you can have a helical capsid or you can have an icosahedral capsid. And this we already been talking about icosahedral ones. That's kind of what I'm drawing up here. It's the ones that you're typically used to seeing that look ridiculous. So you can have naked or enveloped helical capsids. If you are naked, then you're just going to be your nucleic acid just like along the line, and you're just spiraling protein around it. Okay. You can take that structure, potentially that sort of buildup, and kind of squiggle it around itself. Like if you've got this nucleic acid covered, this protein covered nucleic acid, and then you can just ball it up and shove it inside of a membrane. That's what we get with the enveloped helical ones. So instead of having this in here, it will be a bulb of protein around nucleic acid. You're always going to have the protein around the nucleic acid. That's the part that is protecting the nucleic acid. Okay, so that's the capsid. It always has to be there. All right, so these are the icosahedral capsids, which is kind of what I was trying to draw on the board. But yeah, I mean, they do assemble in a sort of icosahedral fashion, just naturally on their own. And you can have naked ones like we drew up here. And then that spikes will be attached to the capsid, and then the enveloped ones, which are probably the ones we typically think of when we think of a virus. So things like it's talking about hepatitis B on the left, and on the right, herpes. And we know that flu falls into this category, as does COVID. A lot of them have that. Okay. So let's talk about some more complex capsids. Y'all tell me this doesn't look like straight out of outer space. So these are the bacteriophages. And that is an actual electron micrograph, an actual image of what they look like. So bacteriophages are viruses that infect bacteria. And you'll typically hear me calling them phages, okay? Because there's no other kind of phage, it's only bacteria. And each bacteriophage infects one kind of bacteria, like only E. coli, for example, and it isn't going to also infect salmonella. So that can be also used as a way to sort of figure out what bacteria you're dealing with in an infection. If you have a phage that can only infect E. coli and your patient has, diarrhea, and you don't know if it's E. coli or salmonella, you could potentially see what happens when you put it with a bacteriophage that infects E. coli and see if you get a response or not. So that's useful for that sort of stuff. But yeah, anyways, so bacteriophages themselves are ridiculous looking. And they take shapes that are not necessarily symmetrical. They look like, look at it. So you've got that sort of icosahedral capsid head that we've been talking about this whole time. And then coming down, you see the collar, and then the sheath that looks quite a lot like a spring, and it is a lot like a spring. Down at the kind of butt of it, down here, you can see it on the electron micrograph, a real life one. This is our tail pins, and then these are our, let's say tail fibers. Legs, right? So these guys will come, and on their tail fibers, that's what will recognize, like that's what has the key on it. That'll recognize the cell, and the cell will unlock for it, right? So it says, okay, this is E. coli. We're good to go. when it docks and it recognizes it as its target, it's literally going to cause it to go through a chemical reaction to squat down. When it squats down, these tail pins are going to stick into the surface of the bacterial cell and anchor it there. And then it's literally going to use this spring to inject the nucleic acid into the bacteria. Isn't that what? So that's how it works. It just injects it in. It doesn't, no circumstance do we see the phage itself getting into the bacterial cell. It always works like this. It just injects its DNA or whatever into it. And I think they're always DNA. I might be wrong about that, but yeah, so it's just going to inject its nucleic acid, its genome, straight into the cell. It doesn't even itself enter the cell. It just, that's its injection mechanism. And when that gets in there, the bacterial cell will start making these guys. These guys, you can tell that they're not enveloped, right? They don't have a membrane around them. How would you get it? How would that work? So it doesn't. They don't bud out of these cells. They literally just burst out. They will replicate until they burst out of the cell, and then they'll go back to the next cell. So that's how bacteriophages work. Pretty brutal and cool at the same time. But yeah, here's some pictures of some viruses, whatever. So the viral envelope, like we said, typically is the membrane of the host. It can be, okay, I'll say this. It can be the cell membrane or the nuclear membrane. The virus that I studied actually would steal the membrane from the Golgi apparatus. So it goes through that whole process of what we would say the packaging and getting ready to transport and everything. And it would just bud out of the Golgi. And then it stole that membrane on its surface. So it's pretty crazy to think about that. But yeah, so anywhere there is a membrane, it could potentially steal it. Most commonly, we think of the cell membrane. If you are a DNA virus, you have DNA as your genome, and you're infecting a eukaryotic cell. Where are you going to have to go in order to start your replication and everything? You have to go to the nucleus, because that's where all of this stuff is for replicating DNA and all of that. So that is why some of them will bud through the nuclear membrane. So yeah. And I'll show you some pictures. I'm going to show you guys some pictures of viruses that infected cells. It is like terrifying, I feel like. But all right, so you either have DNA or RNA when you're a virus, not both. Our cells, do they have DNA? We have DNA for our genome, right? We also have RNA just doing work, like those are working copies, right? Same can be said for the bacteria. Viruses are only going to come with one, DNA or RNA, and it has all of the genes that it needs to build its own virus. Sometimes it'll steal components of the cell and pack it into their virus so they can start work early, but very few of them do that. All right, so we have different kinds of genomes. They can be RNA or DNA genomes. So it's their actual genomes. You can imagine if you are a RNA genome and you're like probably like you could just read it like with your ribosomes and whatever, like we kind of introduced, that would be useful for the virus. Yeah, you could just unsheath your RNA from your capsid, and then the cell can literally just start reading it and making protein right away. You don't even have to do anything else to it. So that is quite useful for a lot of these viruses. So we can have positive sense RNA or negative sense RNA. When you can put a ribosome onto your RNA and just read it and make your protein and nothing's weird, that's positive sense. If you have to make a copy of your RNA first and then that copy can be read, that's negative sense. Okay, so think about it as like DNA. We have the complement, the two complementary strands. What if we made a copy of the other strand that didn't, code for your mRNA? What if that like couldn't read it? We had to make a copy of it first. So it's a complementary copy of it. It's just like the A to U and the C to G thing. That's all it is. So negative sense RNA, we have to convert it to positive sense in order to read it and make protein. That's all it is. I'm not actually going to ask you guys to explain these, by the way. I just want you to kind of get an idea of the differences that we have going on. Do know that viruses have positive sense RNA or negative sense RNA genomes or DNA, double-stranded DNA, or whatever else, single-stranded DNA. Are we even going to talk about those? Okay, whatever. Some of the enzymes that can be coded for by the virus will include their own polymerases, replicases to copy RNA directly, because we don't copy our RNA, right? We make it from the DNA. So they have sometimes have replicases to copy their RNA. So they can just do that and not have to go back and forth. And then some of them have something called reverse transcriptase. Reverse transcriptase makes DNA out of RNA. The central dogma of biology is that you go from your DNA, your Smithsonian institution, to your working copy mRNA, and then that goes to be read by the ribosome to make protein through translation. That was how we always thought. We didn't think you could ever go back the other way. Well, HIV slapped us in the face with this realization that you can, in fact, go back from RNA into DNA, because HIV has an RNA genome, and it comes with reverse transcriptase that will then allow it to convert its RNA back into DNA and then insert it into your genome. And that's how HIV hides out. It's part of the reason why you can't cure HIV all the way. You always have it hiding out. And it's like, for now anyways, right? It's the same thing with herpes. When they say herpes is forever, that is the same kind of concept. It's going to hide its genes in your genes. How do you get that out? You can't just kill the virus, right? Because it's still got its genes in your genes. Who finds that? How do you get rid of that? Likely in the future, we will see things involving genetic engineering to like help silence those genes or help us find them with CRISPR, clip them out or whatever it is. But right now we are not capable of doing that. And so if you have an infection that it inserts itself into your DNA, you're going to have it forever for now. Always. All right, so arenaviruses pack ribosomes, and some of them pack tRNA. Again, that's just to help them start their building before they get into the cell, which has the actual like, nucleic acids, and it has the ribosomes, and it has all this stuff that it needs to do everything. They have some weird ways of classifying viruses that I'm not ever going to ask you. So these were the questions we were going to ask in class. Why aren't viruses considered to be alive? When we talk about viruses not being alive, they can't replicate on their own, right? They have to steal another host cell machinery to do it. So that's one reason they can't live outside of a cell. What are two essential components of the functional virion? So a virus is just virion is a functional virus, yes. Capsid and nucleic acid, that's it. Capsid, nucleic acid. I mean, of course, on these ones, they have a nucleic acid, but I don't want to get into that right now, but yeah. So viruses that lack an envelope. If we do not have an envelope, we call them naked. What types of genomes can viruses have? Well, we talked about RNA. We talked about it could be positive or negative, and it could be DNA. It could be double-stranded DNA, like normal, but we would think of normal. Or it could be single-stranded DNA as well. So it could be single-stranded DNA, positive or negative sense. So what is reverse transcriptase? It's an enzyme that lets us go from basically RNA back into DNA, so we can insert it into a host. And we can use that obviously for genetic engineering purposes as well. Okay. The steps. Okay, the steps of viral infection. So I would definitely know these words, and I feel like they explain themselves as you learn what a virus is and how it works, okay? So know these in order and know them. So, all right, so we have, like, if I were to say, what comes after penetration in a viral life cycle, you should be able to tell me it's uncoding, right? That's what I mean by know them. All right, so we have adsorption, penetration, uncoding, synthesis, assembly, and release. Go figure, we're gonna go through each of these. So adsorption, not absorption. We're not absorbing like a liquid with our paper towel. We are adsorbing, which means we're sticking to something. So we are using the proteins on our spike of whatever it is attached to get into a cell. So when we attach to our receptor, when our key finds the right lock as a virus, then that's adsorption. All right. Host range is usually going to be defined by what molecules you can bind to on the surface of a cell. So if you can only bind to, like, let's say, okay, restricted host range, hepatitis B only infects liver cells of humans. It doesn't infect liver cells of all primates, just humans. So there's something about a molecule that it interacts with that is only found on those cells. So it interacts with that. But if you go down to the bottom where we see rabies virus, broad host range. Rabies virus infects various cells of all mammals. There's some protein that is found on many cells on all kinds of mammals that it uses as its way to get in. Does that make sense? So whatever that lock is that they're putting their key into, that lock It's just some sort of normal protein on the surface of these cells. Is that protein rare to that situation, or does everybody have it, or whatever? So that's what determines host range. Okay, and we know that about rabies. We know that rabies, any mammal can get infected with rabies. All right, endocytosis. So this is our uncoding. We've got, because we're going to go through penetration and uncoding. So we adsorb, we stuck onto the thing. Now we're going to penetrate. So the cell on eukaryotic cells, they typically, whenever you interact with something on the surface, they're going to endocytose you. So take you inside of the cell, right? Endo into cytosis cell. So the cell is going to eat you kind of up, take you into a vesicle. There's often enzymes in the vesicle that will help dissolve the envelope and the capsid to help release the genome into the cytoplasm. So that's usually what's going to happen if you're being eaten up. Okay. Another option here down in B, we are covered in our blanket, right? Our envelope, and we can come and dock with the thing that's on the surface. And instead of being eaten up, we can now merge our envelope, our blanket, with the overall blanket of that cell, that target cell, and then release the capsid and its stuff, all this stuff that's in here, release that into there. Now, oftentimes that capsid's going to get broken down and we'll release the genome that way. So there's two ways you can go in. I'm not going to make you explain them, but just you can understand the cycle and why they happen the way that they do. That's what I'm wanting you guys to get out of it. Okay, so then viral nucleic acid is going to take over the host synthetic and metabolic machinery through synthesis step. All right. We're going to replicate our viral genome and create proteins to help build our genome. So those capsomeres, we're making those and building them up into capsids. Yeah, so it depends, like it says, on whether you're an RNA or DNA virus. Like we were mentioning, if you're DNA, you're going to have to go into the nucleus to do it. So this is the RNA virus. We're going to be going into binding on it outside a cell. Here's adsorption, right? Number 2, penetration and uncoding. We're penetrating. We're uncoding here. We're getting our genome into the cell. So we've got our positive sense RNA here. We can just start reading it with ribosomes. And then typically what you're going to do is just start making capsomeres. You can also make a negative sense RNA as a template to make more positive sense. So whatever. So we're making more genome. We're making the proteins to build our new virus, and our spikes are going to stud the outside of our cell. You can see it building up on the outside there, so that whenever we get the capsids, capsomeres to assemble into a capsid around our new genomes, right, we can actually assemble those, assembly, and then push our way out of the cell. When this happens, when we're budding out, like I said, like when we're covering ourselves with that membrane with the spikes on it, then that's called budding. But then there's another way to do it. If you are naked, you're not going to bud out, right? So how are you going to get out? Well, you just build your stuff to this point right here, and typically the cell will exocytose you. So just like it would a toxin or waste or something like that, it'll create a vesicle and then like push that stuff in the vesicle out. And so that's how they would get out. They don't usually burst out of eukaryotic cells. That's thankfully, right? So yeah, so that's how RNA viruses do their thing. With DNA, they gotta make it extra. So we will take our viral DNA here and put it into the nucleus, and then we can make our RNA, mRNA from the DNA to start making proteins with our ribosomes. Cool. So we know how this process works. We've talked about that already. Now this is something we know. All right, so you can also start replicating the viral DNA. You can use enzymes to just do that on its own, or you can insert it into the host DNA and just keep it there and chill. Either way, they might be doing both at the same time. So we get our new genomes and we get our capsids that have to be brought back in because they were outside doing that, right? We know translation happens outside. So we'll bring those capsid parts, those capsomeres, back in and assemble them around that viral nucleic acid genome or DNA, right? And then it can bud out of the nuclear envelope and cover itself that way. Okay, so this is an image of a cell where this is our nucleus of a eukaryotic cell, and we have all of these little dots here. So this whole thing is a nucleus. First of all, your nucleus should not ever be this big. That's nuts. Second of all these dots here are adenovirus. Adenovirus causes just normal cold symptoms and stuff. It's nothing crazy. It's also what they use. They can hollow out adenovirus and put other genes or genomes into it, and then put that into your body as I'm trying to say vaccine. So you have the two kinds of the COVID vaccine. You have, one type that is like a vesicle contained messenger RNA, and that's all it is. And then you have one that's adenovirus hollowed out, and it contains the mRNA, and that's what that is. And so that can have that adenovirus infect you and create the cell parts for, or create the parts for COVID virus, and then you react to that. You imagine how the vesicle thing would work if I put just mRNA into you, inject it into you. Your cells would then just start making the mRNA protein, right? So if it's COVID surface protein, your cells will be expressing so the COVID surface protein, but your body's still reacting to it as foreign, and that's how that one was made. Won the Nobel Prize. So right, so look at this. So this thing here is all of our viral proteins that are being made and put into the nucleus. They're just like getting backed up to make new virus particles. Can you imagine how this would affect the cell? I mean, this is going to take over everything that's going on in the cell and cause like just serious issues with the cell being able to function. And that's exactly what's happening in your cells. It's going to cause that kind of a damage. And yeah, of course, it's going to make you feel like crap. So when we're releasing our mature virions, we're basically pinching off into that little pouch that we are pushing out into. It's called budding, or if you're not budding, you can exocytose, be released by the cell through its normal mechanisms. All right, so damage to the cell. These are our cytopathic effects. These are anything that's happening as damage or prohibiting the cell from working normally due to infection by a virus. That's a cytopathic effect. Okay, so this will include things like the cell becoming disoriented, undergoing changes in shape or size, developing intracellular damage, inclusion bodies. Like it's kind of like what we saw here, the massive amounts of protein. We'll see a lot of those in cells, just containing whatever it is that this virus is making. And then syncytia. A singular would be syncytium. So this is fusion of host cells into single large cells containing multiple nuclei. So if I'm telling you that this spike protein here is going to get studied on the outside of your membranes, right? So we can bud on out and make a new one. All right. And you're next to another cell that you may or may not be making virus. It doesn't really matter. But you've got these that are the key into the cell next to you, studied all over your membrane. Of course, you're going to have cells merging to each other because they're going to be recognizing that, oh, yes, we merged together now because this protein binds onto here. And that's what we normally do. So they think that this is normal, but it isn't. So that's syncytia. So cells merging to like the guys next to them and creating one big old massive multinucleated cell. If you've heard of RSV, respiratory syncytial virus, guess what it does? Forms syncytia. So then cells aren't able to function the way that they're supposed to. They're not meant to be one big old blob of cell. They're meant to be separated for a reason. It's part of why the structures of your organs operate the way that they do. All right, persistent infections. You can carry DNA in your genome, like I've mentioned. Stick that DNA into your genome and you have it for life. That when it does that, that's called a provirus. It's not a true virus, right? It's still just the genome shoved into you, but it could make a virus. So it's provirus, like you can think of it as before. The virus is going to be made later. So they can hang out like that for a long period of time. There are a lot of viruses that do this, a lot, way more than you probably think. We're talking about chickenpox. We're talking about EBV. What is it called? Mono. That's what it's called. Mono, HIV, herpes. All of these are going to be guys that will insert themselves into your genome and just hang out there for the rest of your life. And we know this about chickenpox. We just don't think of it as any big old deal because, yeah, you just have it forever. And you can just turn into shingles later on in your life. And we don't really think about that. But what it's doing is hiding in your DNA because chickenpox, if you didn't know, is a herpes virus. So all the herpes viruses do that. Same thing with mono. It's also a herpes virus. So yeah, think about that next time. All right. And then we know about the oncogenic viruses. You guys know what onco means. That's going to be cancer. Oncogenic viruses are viruses that cause cancer. We think about 13% of our human cancers might be caused by viruses. Not all of them have been discovered. So transformation is a term, when we're talking about viruses, okay, because we'll talk about genetic bacteria doing transformation and stuff like that. But like when we're talking about viral transformation of a cell, like a eukaryotic cell undergoing transformation, that means we're inserting genes into the genome of that eukaryotic cell and making it cancerous. So it will multiply out of control, unchecked in the cell cycle. Normally your body will like say, hey, you're multiplying too much, maybe don't do it. And yeah, it doesn't in these cases. So the virus is going to interrupt that cycle. So we can create transformed cells or, tumor or cancer cells. Why would you want to create tumor or cancer cells? It's not necessarily that it helps you study those exact things, but it helps you generate an immortal cell line that you can then use for studying human diseases without having to use animals or sacrifice any sort of, person or fetus or whatever it is that you might want to think of. So that can be helpful for them. All right, we already know about the bacteriophages. Most of them are double-stranded DNA. I'm not ever going to ask you what kind of genome they have because I didn't even know. But yeah, there you go if you're interested. So these guys are going to, again, squat on down and inject their nucleic acid into the bacterial cell. And other than that, the stages that they're going through are pretty similar to what our viruses do, the naked viruses anyways. And then here's an image with a cell just coated in these bacteriophages. All right, so when we're talking about something like a T-even bacteriophage, which I'm not going to ask about T-even, right? But I might ask you about a temperate phage. A temperate phage is a phage that can cycle between the lytic and the lysogenic state. Okay, so the lytic cycle is what we've been talking about. So you infect a cell, you make more copies, and you burst out. Go to the next cell, infect it, make more copies, burst out, whatever. That's the lytic cycle. The lysogenic cycle, I think of this lysogenic, maybe this is weird to you, but because this is the gen, I think of gene. This is the one inserted into the genes of the bacteria, okay? Lysogenic state. You're putting your prophage, when we talk about provirus, that would be put into your genome, prophage for this, okay? The prophage is inserted into the bacteria. And now when the actual bacterial cell that has that divides, it's going to be making copies of that. Same with your cells if they're infected with whatever, but here we're talking about this. So lytic phase, normal, just replication of the virus and ends in the destruction of the bacterial cell. The lysogenic cycle will have that prophage being made, and then we get it replicated with the cell DNA when the cell divides itself. So these are temperate phages. They can switch between. So think of temperate as kind of in the middle. You're not leaning one way or the other, right? You can go in between. So our prophage is our inactive phage DNA in the host chromosome. Induction is whenever you are going to go to that lysogenic, where it's incorporated into the gene, and something happens that causes it to switch back into making viruses again. So that's the induction. All right, there's also an issue with lysogenic conversion, probably one of the most important things to be talking about if we're going to talk about phages. This is whenever we acquire a new trait from our temperate phage. So you are a bacteria, and I don't know, let's say you are, let's think of one that's good. You guys heard of diphtheria? Diphtheria is part of the TDP vaccines, right? The tetanus and diphtheria and pertussis. Anyways, diphtheria is a severe respiratory disease. It creates a pseudomembrane in your throat that can actually block off respiration entirely. It's gross, but we don't see it really here in the US because of vaccination and people keeping up with it. Well, Corynebacterium diphtheriae, which is the bacterium that causes diphtheria, it has been lysogenically converted. What that means is it was just a bacterium, Corynebacterium diphtheriae normal, and it was infected by a phage that became a prophage and inserted genes into it with it that carried a toxin gene. So now Corynebacterium diphtheriae has that toxin that it produces that actually results in having, true disease for diphtheria. It's so significant for that organism, for diphtheria, for example, that when we vaccinate for diphtheria, we vaccinate against the toxin, not the bacteria. Same thing with tetanus and pertussis. And you would be surprised to learn how many bacteria do in fact have this situation, the lysogenic conversion. Yeah, if I say cholera, man, the whole reason cholera makes you have rice water stools, which is literally out of your bottom, rice water. It's not brown anymore. It's just you've lost so much and you're losing so many electrolytes. It looks like whitish water coming out of your rear. The toxins that cause that release of your electrolytes from your cells in the gut, they came from lysogenic conversion. So just from a virus that infected a bacteria. Nuts to think about, but there you go. All right, tissue culture. If you're going to grow viruses and study them, you need to have cells for them to infect. They can't be grown by themselves. So if you're studying eukaryotic ones, you will need to have a cell culture that you can propagate to grow your virus in and study it that way. Maintaining those cultures is probably like one of the biggest pains in the butts for virologists of all time. Yes, so we don't know for sure the amount of viral infections worldwide because honestly, some of them aren't even, discovered yet. I mean, really. And by some of them, they're going to cause like a mild cold. You go to the doctor and you just have a cold, and your doctor's going to be like, "Yeah, you have a cold, man. Just go home." Like, do they test you for the viruses causing it? Not usually. If you're a kid and you're having severe symptoms, maybe. But like typically, if you just have a cold, you might not even go to the doctor. You just be like, "Oh, okay, well, whatever. I'll just wait a couple days. I'll be fine." And it's probably true. But do you know for sure that it was like rhinovirus or adenovirus or something else that's really common for causing a cold? No, it could be any random thing that just like popped up or has been in the population that nobody's really just discovered that can't get it to grow in tissue culture, for example. Who knows? Some have high mortality rates, like rabies and Ebola. Ebola, we already know, causes you to bleed out, hemorrhagic fever. Basically, the cells, when they are infected with the virus, they don't adhere to each other the way that they're supposed to anymore. It's a cytopathic effect. And so on the epithelium of your blood vessels, it becomes weaker to become very fragile blood vessels. And that's what leads to the hemorrhaging of hemorrhagic fevers in general. So that's how that will work. But it's crazy to think that like one little tiny virus can do that to a whole body. But then rabies, you guys don't underestimate rabies. Don't underestimate rabies. It's not a joke. You might think like, okay, well, we don't really have this, but we do. It's rampant in like wildlife. So I think here it's skunks that's the biggest issue. Skunks, bats, and raccoons tend to be the biggest carriers of it. It's not actually typically the coyotes or anything like that, or wild, stray dogs or anything. It tends to be those guys. But if you aren't vaccinating your pets and they get in a fight with a raccoon or something, which would definitely happen at my house because we have so many friggin' raccoons and stuff. I don't know how, but they're all the time in our backyard. You get in a fight with a raccoon and it bites your pet, and now that raccoon that was rabid, you didn't have your pet vaccinated for rabies. All of your other pets were vaccinated. You thought it would be fine because, herd immunity, basically. That pet gets bitten. That pet will not immediately. It will take quite a bit of time, but they will develop rabies and die. So that's serious enough. But what happens if you've seen like these videos of like raccoons running up to people and like attacking a kid, and like the guy like throws the freaking raccoon off and whatever, or throws the kid and runs off or whatever it is. Anyway, what happens if a rabid raccoon runs up to your kid and bites your kid? Now you might think, okay, this scary raccoon, maybe it bit my kid, and you'd be smart enough to take it to a hospital, and they would start in with immune globulin and shots for rabies, and you'd be like, it's fine, it's totally fine. Yeah, if you do that, it's fine. If you go in there and you get the immune globulin, that is already stopping the body from letting the virus get into the cells. You have to have that. You have to have the vaccine itself so that when the immune globulin isn't there, you have your own, immune globulins to protect you, right? And that's fine. But if you just let it go, what are the odds that, let's say that your kid gets bitten by the rabies and you didn't take him to the hospital and he got rabies, right? What are the odds that your kid's going to die? Do you guys know? It's 100%. Mm-hmm. Dead serious. It's 100% mortality rate.
When the raccoon bites a human, is it still called rabies?
If it's only if it's rabid, right? So if it's just a random raccoon, whatever. But if it has the virus for rabies, it's actually rabid, and it bites you. It tends to be the way that the rabies virus works. It causes the host to salivate a lot, which is why you see the foaming at the mouth. And the virus is actually found mostly in the saliva, so it will like be expressed there. And so it's like an advantageous trait that rabies has. So that's why the biting is the real issue, and not scratching or something like that. It's the biting, it's the saliva. You can even have passage of rabies without biting if like your pet like drank from a bowl that a rabid animal drank from and had open sores or something. It's not just going to randomly get in. But yeah, rabies is 100% fatal. If you get it in your body and it actually has infected you, you will die. 100%. So that one's not as bad. So that will be the toxin for staph, right? For rabies, for this virus, you usually won't see symptoms in humans for a month to 2 months. So yeah, you might think your kid's fine. Yeah, to actually get that prevent it. I don't know like what the exact time frame is for that, but I feel like it is. You want to do it within a week. But yeah, I mean, if you do get bitten or a kid that gets bitten, I mean, it's not going to be like minutes. Yeah, you need to get them to the ER and get them treatment, but you'll have time to do it. Anyways, yeah, I always try to tell people about that because I feel like it's like so underrated because we vaccinate our animals. Don't take it for granted. This is really, really serious. You don't want someone to have rabies. Okay, so we're pretty much done. I feel like that's it, right? Interferon, just FYI, is a cell protein that signals the next cell that there's an infection. So it's like if you were, getting robbed in your house, there's a really bad, serial killer and he's, robbing your house and whatever, and you hear him and you can't leave, but you have your shotgun and you're hiding out in your closet and you call your neighbor and he's like, there's this guy, he's coming, you need to get ready. And like, you might die, but your neighbor has their shotgun already out and ready to go before that guy even shows up. That's interferon. So it lets the cell, the next cell know that there's a viral infection coming, basically. So it's pretty cool there. And then real quick, prions. Prions are the proteins that are misfolded, that because they're misfolded, normally we're going to be hiding away our hydrophobic guys, misfolded now. So they're not hidden away anymore. So they're going to clump together typically and cause holes to form in the brain tissue. And that leads to something called spongiform encephalopathies. Spongiform because it looks like a sponge, the holes that are forming because of these misfolded proteins. And then encephalopathy is just disease of the brain. So that's prions. This includes CJD. It could be variable CJD. It could be inherited CJD. But there's all sorts of different kinds of Creutzfeldt-Jakob disease. And we also have other diseases in animals like scrapie and bovine spongiform encephalopathy, which is mad cow disease. And in humans, we also have Kuru, which is where cannibals eat each other and they get it. Yeah. And then fatal familial insomnia is also one that falls into this category. These are super rare, and they're just proteins causing the disease. It's just a misfolded protein. And when it comes around to your normal version of the protein, it causes your normal version of the protein to turn to misfolded. And so that's how it propagates. But it's not got we don't know how it started. We think that that's the way that they're propagated once they get into the brain tissue. If you eat, if you were in Britain and you were eating mad cow disease meat at the time, then 30 years later, you may have gotten this. Because we're talking 30 years later, you have this horrible disease that's like within a year, you will die once you start having symptoms. Again, 100% fatal, these guys. There's no treatment for these. So yeah, that's that one. I'm not going to go over the questions this time. I don't want to deal with it. So don't forget those proteins. Prions are only proteins. There's no genome there. Just a misfolded protein. Yeah, we should be fine as long as everybody's like aware of what everybody's role is and working together.
Yeah, they're doing the presentation. I'll plug in my microphone to my laptop and it shows some pond water. I don't want to show the pond water, but that's not really what we're going with.